coming. Just want to introduce Dr. Kristen Park and Katie Angione talking about genetic epilepsy in pediatrics. So a brief introduction, Dr. Kristen Park grew up in Baltimore, Maryland, and currently lives in Denver, Colorado. She attended UNC Chapel Hill for her undergrad studies, attending a BA in psychology. She attended Mayo Medical School and did her pediatric and neurology residencies at the Children's Hospital of Philadelphia. She then moved to Chicago for her epilepsy fellowship. She finally got her first real job, folks, at Children's Hospital of Colorado, where she is an associate professor and director of the Epilepsy Fellowship Program. Her main clinical and research interests are in neurogenetics. Katie Angione is a neurology genetic counselor at Children's Hospital Colorado. She provides genetic counseling for a diverse population of patients with complex neurological phenotypes, with a focus on developmental and epileptic encephalopathies, or DEEs. Katie works with patients and families with rare disease in general neurology and epilepsy clinics, as well as Rett Clinic, Neurogenetics Clinic, Tuberous Sclerosis Clinic, and a new multidisciplinary clinic at Children's Colorado serving patients with, bear with me, STX-BP1, SLC6A1, Ring 14, and chromosome 8p disorders. Katie's primary goal as a genetic counselor is to support these patients and their families through education, advocacy, and research efforts focused on understanding the natural history of these conditions and eventually working toward precision diagnoses and treatments. Without further ado.
Thanks everyone for joining.
All right. Don't applaud yet, we haven't said anything. So thanks for coming, everybody. As Kristen said, we're going to talk a little bit about genetic epilepsies. We have a prepared set of slides that just highlight some things that we wanted to go over, but also I think this is a relatively intimate setting, so we can also do a lot of Q&A, which we had at the end, but I think we'll kind of breeze through some of the hardcore stuff so that we can hear from you all and field some questions. So Katie's going to start. We're going to try to advance this as we can. She has it on her computer, so she doesn't have to look back, and crane her neck. So,
all right. So we're going to kind of go over a few different things today about precision medicine as it relates to genetic epilepsy disorders. So we'll start with a little bit of background, go through kind of one of the more new specific precision medicines. Dr. Park's going to talk a little bit about SCN1A and some lessons we've learned from precision medicine there. And then we'll kind of go over what's going on overall with precision therapies and kind of what we're looking at for the future. And then we'll open it up for questions. And like Dr. Park said, we're going to go through some of this a bit quickly, so don't feel too overwhelmed by that. We just want to get to the end and be able to answer the questions that you guys have.
Do you want me to do this?
Yeah, it's okay. I got it. I can multitask. So our little video is not working here, but we wanted to start with just a general overview of how genetics works. So basically, we all have DNA that makes us who we are, makes up how our body works, everything that kind of functions to aid in our growth and development and the functions of our brains and our bodies. So the DNA is kind of like the recipe. It's the starting material. It's supposed to be picking us up, but maybe it's not.
I think that's for the recording, so
okay.
Maybe it's not loud enough for them to hear out there.
Is that better? No, there we go. Yikes. All right. So the DNA is kind of like the recipe. It's the starting product that everything in the body is built from. So when you look at a genetic testing report, you see all those letters and numbers. A lot of that is just explaining changes within the DNA, so within that recipe. The DNA is then used to build proteins. So our body takes the DNA, it makes it into RNA, which is sort of the in-between part. It's the messenger that goes from that code and takes it into the final product of the protein. And then the proteins are the things that are helping our bodies to function the way that they're supposed to. So protein is not what you're thinking about when you think about, getting your protein in for the day. Protein is just anything within the body that's serving a function. So that's what we're looking at when we're trying to figure out how to alter a genetic disease is, can we alter that beginning recipe of the DNA? Can we do something to change any problems with the RNA, the in-between messenger? Or can we make a change in the final protein and correct any issues that are preventing it from working the way it's supposed to? So when we think about precision medicine, it's important to understand kind of how this process works and all the different points along that process that we can target to correct any errors or any issues that are causing genetic disease. It's gonna work. There we go. Very good thing to remember anytime we're talking about developing therapies and treatments. Mice are not people. So mice, zebrafish, all the different models that we use to investigate different therapies, they're not perfect models. So we do our best, obviously, when we're developing drugs and therapies to try to understand how they work. But there's always going to be barriers that we run into along that process. So that's why it takes so long for us to get from understanding a disease to figuring out targets for precision therapies to actually getting to a drug that we're able to test and hopefully eventually use on humans. Where are we? All right. So we have a lot of different goals with precision medicine throughout that process. So first of all, it's really important for us to be able to characterize the phenotype or what that disorder looks like. What symptoms are we seeing? What parts of the body are impacted? And that can take a long time when we're talking about rare disease because we're working with small groups of patients. So that's kind of the starting point, is understanding the natural history of disease and what things look like without any medications or therapies. Then we work with, there's a lot of different ways that we model the condition to understand how they work in the body, and that can be through animal models, can be kind of petri dishes. We're at the point that we can sort of make little tiny brains in petri dishes and see how different changes affect those neurons and those cells. We can look for biomarkers, so things that can be measured throughout the process of research to see, are we impacting the course of that disease? Are there things that are changing if we're applying these therapies? And then targeted drug screening, so looking to see, are there specific medications that work better or maybe are not good to use for a certain condition or a certain gene. So we have, and I'll put up here in a second, we have a list of medications or a list of genetic diseases that we have figured out over time. This medication works really well. This is one that we need to avoid. So that's a big part of precision medicine in epilepsy because, as a lot of you probably have experienced, there's a lot of trial and error when it comes to epilepsy drugs. So part of precision medicine goals within epilepsy are trying to fine-tune things a little bit more to kind of move through that process of trial and error a little bit more smoothly for some conditions. Obviously, we want to consider the genetic mechanism of disease when we're working on therapies. There's a lot of different, like I said, a lot of different points in the process of DNA to protein that can be targeted. There's also a lot of different types of genetic mutations, and those can be targeted in very specific ways. So depending on the exact change in the gene, there might be different ways that we can correct that change, or that we can kind of work around what the body is kind of doing differently or doing incorrectly to change the final outcome. And then at the end of the day, we always have to think about the money and the funding. So it's really important to be able to partner with industry, with family advocacy groups, all of these people coming together, kind of raising money for the research that gets us to the final product. And then once we have those therapies in place, they tend to be very expensive. So that's one of the other barriers and challenges, is working with insurance, trying to get funding and support for families once those drugs are developed. So it's a very long process and lots of steps along the way. So this is just kind of it doesn't look brief, but actually a brief list of the genes that we have precision therapies for at this point that are either pretty solid or are emerging evidence for precision medicine. So this can be anything from specific drugs that target certain genes, to ketogenic diet, to even gene-specific therapies. So when you hear about gene therapy, that can mean a lot of things. But everything on this list are kind of drugs or genes that have implications for precision medicine. So if we make one of these diagnoses in one of our patients, we would talk about, all right, well, now that we know that this gene is the underlying cause of your child's epilepsy, now we have a little bit better idea of how to treat it. So that's just more genes with precision medicine therapies.
All right, so now we're going to change tactics a little bit, and I think along the lines of what are our therapies and what are our options, what do we learn when we make a genetic diagnosis. I think we're going to take some lessons from SCN1A, which is one of the first epilepsy genes that was discovered. It's also one of the most common epilepsy genes. And so that particular organization and diagnosis has made a lot of progress in understanding all of those points that Katie listed about what we need to know to develop therapy. And so I just kind of want to go through the state of things for that one gene, recognizing that it is kind of a model for many of these other genetic conditions that are related to epilepsy in how to move forward and what needs to be done. And these are kind of cutting edge things that have been identified for SCN1A. So SCN1A is, in fact, related to a particular epilepsy in children called Dravet syndrome, where children typically have multiple seizure types that are very treatment resistant. It is a spectrum of disease that it can cause, but this is the most classic form. So over 1,800 genetic variants have been identified for this particular type of epilepsy since it was discovered in 2001. And because it's so common, again, we've been able to hit a number of those bullet points that Katie mentioned. All right, so I think this is one of the newest papers that was published in Epilepsy in 2022. This paper kind of blew my mind when I read it. And basically, this gene was discovered in a large family, most of whom came from Australia. They had multiple generations of people who were affected by epilepsy in some way. And the interesting thing about this family was that not everybody had the same type of seizures. So some people had Dravet syndrome, which was very serious. Some people had no seizures. Some people had one seizure in their lifetime. Some people had febrile seizures. Some people kind of had an intermediate type of epilepsy. And yet when we looked, they all had the same genetic variant in their gene for SCN1A. And we said, how can that be? How can these people who have the same gene that's affected have such different presentations? And so just recently they went back to that same family and they said, let's try and see if we can figure that out. Maybe they have a change that they share in some other gene that modifies how this gene presents itself. And what they found was that they actually had a second variant in that same gene. And the people who, and when they expressed those in a model of epilepsy, I think it was HEC cells, it canceled the other one out. And so people who contained both didn't have very serious epilepsy or had no seizures. And people who just had one had the very severe epilepsy. And that is just huge, huge in the epilepsy genetics world because it opens up a big understanding of why that might exist in other genetic epilepsies. There are these variants and how can we find them? Of course, this is a really large family with multiple people affected, and that's very uncommon in genetic epilepsies, but hopefully as we pool together our resources, we may be able to identify those modifiers in other genetic epilepsies. So the next one that I thought was really interesting that I wanted to highlight, again, came out in 2022 in neurology, was this is a big question. How do we figure out whether a variant in a gene causes disease? So if we think about our genetics and we compare the genetics of each person in this room, there are going to be about 3 million differences in the spelling of our genes. And our task as the doctors is to figure out which of those 3 million differences just makes us different from each other as people, and which of those 3 million differences makes us have a risk for a condition like epilepsy or high blood pressure or diabetes, something like that. And that is a challenging thing to do. And it's sometimes not as challenging, so if we use, if we think, well, this change in this gene is going to make it stop in the middle and not make a whole protein, it's not going to work. Well, it's pretty easy to understand that gene, that genetic change may certainly cause disease. But if it just switches one letter, that's much harder to figure out. And so this recent paper looked at SCN1A in particular and tried to use automated intelligence to make up a score that could figure out which of the changes in that gene were going to cause problems and which of them were not. And they combined that with the age of onset of seizures and were really accurately able to predict which of the things that they found was going to cause disease. And that's also great because that allows us to be more accurate, predict what the course of things is going to be earlier. It also allowed prediction within the spectrum. So it said who's going to have more severe disease, like the patients who have multiple seizure types that are drug resistant, and who might just have febrile seizures. And that's really important if we're going to think about doing genetic therapy early. Are we going to take a lot of risk because this is going to be a really serious outcome, or do we want to not take too much risk at all because this is going to be a more benign outcome? So that was the second one I wanted to highlight. The third was this critical window process. So if we think about genetic diagnosis, it happens after someone is already born, after their brain has been formed and they have their first seizure. And a lot is happening, and there's kind of this concern in precision therapy that there's a critical time point before which this type of therapy must be delivered as the child is growing, and typically in infancy, as things are kind of wiring and forming. And so this particular study actually looked at, again, mice who were older. And so the mice were, I think, like P30 and P90, which is like a child and an adult and a teenage mouse, per se. And they gave them these therapies. And I think this is on the next slide, maybe. So P30 is four human years, and then P90 was about 20, so adolescent mice. And they gave them this precision therapy that they had developed and found that they were still able to have an effect at those time points. So even if somebody doesn't get their genetic diagnosis until, kind of several years into this journey, there may be still ability for precision therapy to be effective. Again, kind of huge. Still mice, but huge. The last thing I wanted to highlight was that I think part of the new kind of progress that we're making in epilepsy research is realizing that our brain has the same tools all the time, whether the condition that we present with is epilepsy, or whether it's MS, or whether it's depression, or something like Alzheimer's. The brain only has so many things that it uses to grow, to communicate, to process information. And so there is a lot, now that we're doing a lot more crosstalk between other people researching other diseases, we're discovering shared pathologies. And one of them is tau pathology, which has been implicated in Alzheimer's disease. And they're showing that protein that kind of gloms up inside of the cells when people are diagnosed with Alzheimer's is also present when people have epilepsy. And that just opens up, again, a huge therapeutic avenue that we didn't have before to investigate those therapies to see whether any of them would be helpful in epilepsy and in genetic epilepsies in specific. And this was just one of the experiments that they did that kind of shows that we're thinking about that and trying to work towards it. All right, so this next part is about gene-based therapy, which again, I think is kind of a novel thing that is developing in epilepsy. So the big one that has made the most progress is something called antisense oligonucleotides, or ASOs. and they are being developed for a number of different genetic diseases. Basically, they are a short set of nucleotides that can bind to that messenger RNA and regulate the expression of genes. And there are FDA-approved ASOs. They've been around since 1998. The most recent ones have been approved for spinal muscular atrophy in children, which is another severe neurologic disease. And so they do exist already. We're not as far behind on those. And so there is one that is being developed for SCN1A. This is from Stoke Pharmaceuticals website. So their technique is called Tango, and they deliver these little ASOs in little fat molecules. And basically what happens is that as our body makes these proteins, they transcribe them, and some of them they put in something called a poison exon, which causes the body to say, oh, no, we can't use that one, and we just degrade it. And this particular treatment kind of binds to the regular splicing and helps take that out so that it will produce more of the functional copy. So you already have the gene that is not working. It's going to continue to do whatever it does, and it doesn't work, and it doesn't help people who have that epilepsy. But now we're going to upregulate the body's natural ability to produce SCN1A by preventing half of it from being degraded by this natural process. And so we're going to give you more of a normal copy of that gene. And so that is, I think on the next slide, currently undergoing clinical trials. It is in what's called a phase 1 trial, which is where it's being evaluated for safety and dose in the patients, not necessarily efficacy. So it's a very small group of people. And this company has chosen like butterflies as their motto for all their clinical trials. So their first trial was called the Monarch trial. And then in Europe, I think it was called Swallowtail, and then now in Europe it was Admiral butterflies, and then they're doing the Swallowtail study, but whatever. So what happened was they gave it to they did an interim analysis that was released for AES, and they gave this medication to 21 patients, and about 73% of them had a reduction in seizures, and this was most pronounced in the younger patients, partly because they probably are at a stage of their epilepsy where they have more seizures, but also potentially because of that critical window. There was no safety-related concerns, and they're going to kind of continue to monitor those patients over time. So really promising that this is rolling out in humans and not just mice.
So I think we've hopefully illustrated that it's a very long, arduous process to get to gene therapies, but we do have a lot to celebrate at this point, and hopefully moving forward there'll be more and more therapies available for specific patient populations. At this point, we have available gene therapies for spinal muscular atrophy, which Dr. Park mentioned already, another neurological condition. We also have therapies available for Duchenne muscular dystrophy and for a specific type of Batten disease, which does have epilepsy as part of its kind of presentation. So we've seen patients with all of these conditions at Children's that have had these treatments and have been pretty successful. And it's really crazy, especially to see families that have siblings that both have these disorders. You see the older siblings and kind of how they've done over time without those treatments, and their younger siblings who have access to these treatments, and kind of the difference in what that course looks like. And it's a lot, but it's really amazing to see how effective some of these treatments have been for those families. There's a lot of genetic epilepsy disorders that have therapies kind of currently under investigation now, mostly, again, in mice models. Some of them approaching being able to move towards clinical trials in humans. But there's, like I said, a long list of conditions that we're starting to see a lot of progress in. So we're excited about where things are at and where things are moving. So hopefully next time we give this talk, we'll be able to add some more therapies to that list. I think it's always important to talk about what this means for adults also. A lot of times, especially for us, we work in a pediatric setting, so we're talking about children a lot of the time. We're talking about implementing these therapies as early on as we're able to. But I think it's important to recognize that there is still, it's still, we can still impact the course of disease and how things are going for adults. So it's not just within this critical period that these types of treatments can be effective and are important. We can also hope to improve quality of life and improve disease course and maybe progression in some cases for adults as well. So that critical window is important and is something we think about a lot with these therapies, but we hope that we can also get to the point that we can treat kind of throughout the lifespan and improve things in some way. This is just kind of a snapshot of a lot of the family groups and different groups that we work with that help to push this research forward. So a lot of the funding and research comes through things like family and patient advocacy groups. There's also a lot of different online sort of databases that help to compile all of this information to put it together into something that's useful. For a long time, we've had a lot of different groups and different researchers working on things separately, and I think we have this big push right now to bring that together a little bit more. Like I said earlier, in the world of rare disease, we're working with small numbers of patients. It can be really difficult to make progress and to understand how a treatment might work when you only have a few patients that have been identified. So there's a big push to compile all that information in a way that can drive things forward for a lot of different conditions. So, perfect. We're at 10:30, so I think that was kind of our goal, was to have half the time for questions. So please ask us your questions. We'll try to answer them.
So for people who have similar, different rare conditions, like for example, like potassium channel mutations, have they found treatments that might work for, like my daughter has KCNA2, like a treatment that works for KCNQ2 also be some, or just, they're both potassium channel mutations.
Yeah, so I think that the question is if you have a set of genes like potassium channels, do treatments work across those different conditions? And I think the answer is kind of dependent upon the mechanism. So for channels and what we would call kind of channelopathies, that's a big category of epilepsy. Just to kind of give some general background, channels are how ions or charged particles flow in and out of cells, and that is part of how brain cells communicate with each other. They communicate both electrically as well as chemically with neurotransmitters. And so this is a disorder that affects how that electrical communication occurs within a cell. And this is potassium, which is positively charged and moves in and out of the cell. So there are different types of potassium channels, right? So we have, and they're different because they are expressed in different places throughout the brain. They're different because they have different functions. They're opened in different ways. They turn on they open at different times within the communication cycle. They also can be perturbed in different ways. So some of the potassium, and many of the channelopathies have what are called gain or loss of function, right? So that means when you have a mutation in that potassium channel, that may either make the potassium channel work too well, or it may make it not work well at all, and that would be a loss of function versus a gain of function. And so the different potassium channelopathies have different combinations of those two types of effects. And so some medicines work well for the loss of function, and some medicines work well for the gain of function. And so if we take one medicine that works well and is a gain of function, treats gain of function in one condition, depending on where those potassium cell channels are, where another set of potassium channels are expressed within the brain and what they do, they may need a completely different medication to affect them in a different way. So I think that's a rather complicated thing to talk about because of how different they all can be. What we do kind of use, especially for KCMQ2 and Q3, is we recognize that potassium and sodium channels kind of go together in that spot for that disorder. And so we don't have a lot of potassium channel medications, but we have a lot of sodium channel medications. And so we found that we can affect the sodium channels, which then helps the potassium channels and helps those kids have better seizure control. So I think it's partly a matter of discovering the physiology of what all of those channels do and how they work, and whether we need to make them work better or stronger or less, when and it's kind of a complicated thing. So I think there's not a straightforward answer, but I think that's kind of a good place to think about things. Again, how can we all work together? And there is an organization that we had highlight up there called, it's like International Channel Epilepsy Research or something, where they're trying to group all these channelopathies that have similar mechanisms together so that they can coordinate research and see what can work for one, could it work for another. I think that's this one, the International Ion Channel Epilepsy Patient Registry. So I think they're also doing a lot of basic science research to figure out what medicines might crosstalk between different channels, again in models, but with the hopes that they could be translated towards patients. That was a long answer for a short question, but yeah.
How do you approach your patients in the sense of how do you decide which ones are going to get the deemed testing or which ones are not? Or do you go?
So once we get to that point,
You want to repeat the question?
yeah. So the question was, how do we treat these things with our patients? How do you kind of decide who gets a treatment and who doesn't? It's not up to us, luckily, as providers, kind of which individual patients get access to treatment. But it is
testing too. The testing.
Yeah. Oh, the actual genetic testing.
Testing.
Oh, okay, yeah. So we are pretty lucky, I think, at Children's Hospital Colorado, as we have the ability to kind of get access to a lot of testing for a lot of our patients. It's definitely different from institution to institution how easy it is to actually be able to send testing, because we have to go through insurance, we have to think about, sometimes we have to alter what we would ideally do based on what insurance will cover, and that's really tricky. But we're lucky that there are some testing programs out there that are providing testing at no cost to families. So Invitae is one of the labs that we've worked with a lot over the years that offers a no-cost epilepsy panel for patients under 8 years of age. That's been huge, I think, for a lot of families that typically would have to pay out of pocket or go through a long process to get access to testing. So I think that's kind of the access side of things. As far as who do we offer testing for, who do we want to do testing for, I think the threshold has come down over the years as it's become easier to do genetic testing, as the cost has come down, as the time it's taken to do testing has come down. I think we're more readily offering testing to patients kind of earlier in their disease course and earlier in their lives, because we see the advantages to getting that early diagnosis, especially when we might be making a diagnosis that impacts what medications we're recommending, what therapies they might have access to, what research they might be able to participate in. So there's a lot of kind of reasoning to getting genetic testing and trying to get a genetic diagnosis as early on as we can, because it can change things a lot for those families. And even when there's not, a practical change in treatment because of a diagnosis, we've seen how huge it can be just in, alleviating some guilt or some worry that maybe there was something that happened during the pregnancy, or there was something that could have gone differently. We've had conversations with a lot of families that just having that black and white answer is helpful, even if it doesn't really change anything in that moment when it comes to treatment. It can just be really helpful to have an answer.
So I agree with all those statements. I would also add, just from a practical perspective, so in my mind, approaching a patient with epilepsy, I certainly do genetic testing for anybody who has onset of seizures in infancy, anybody who has a developmental or epileptic encephalopathy, so a very severe type of epilepsy with multiple seizure types and a very abnormal EEG that affects their developmental trajectory. I typically do genetic testing when I have a patient with epilepsy that has no other cause, so their MRI is normal, they have seizures, those seizures are difficult to control. I wouldn't think about doing genetic testing there. I also think about it if they have multiple other medical conditions. I think if they have a family history, depending upon that family history, we might think about doing genetic testing. So I think there are a number of situations in which it is appropriate. And those situations have, as Katie said, expanded over time, and the ability to do that testing has made it more likely to happen. Yeah. Mm-hmm. Yeah. So the question is, oopsie, I turned myself off, sorry. So the question is whether we've seen SETD1A. And I don't know that I have any patients myself who have that, but I certainly have heard of that diagnosis and recognize that association. I don't know if we have any precision treatments for that particular diagnosis at this moment. The panel that we talk about doing for genetic testing has 4,000 epilepsy genes on it. And so we don't really know all of them. And I think we're not at the point that we have those types of treatments for most of them.
Sweetie, just want to share with you, Laura's like 36, and she started having seizures, absent seizures, when she was 3 years old. And so this has been a long progress, and we have seen lots, and this genetic testing is really exciting to us. So she went from being told that she had absent seizures, then when she was 13, it wasn't showing up on the EEG, so they said it was tics. They took her off all medicine, and she had her first grand mal. But and she was always kind of hard to control. So she goes to Johns Hopkins, and so after 5 years, she was going back and forth. She wanted genetic testing back and forth. And as a mom, I was almost afraid, to find out. So she ended up getting the genetic testing, and all the genes that you talked about that we were maybe expected to see, it comes up with this D1A, which is very rare. And we found out too that for some reason Depakote is the only medicine that would hold her. She went through everything. She hates Depakote side effects, but every time she got off it, she would have a brain mal. And so then it came up to, I've got a son that's getting ready to get married. He's 29 years old. So I went back to a genetic, what am I supposed to tell him? Well, since it's a variant of unknown significance, and there's not much to know about it, if Laura was going to have children, yes, they strongly recommend them test it. He's had one seizure when he was 4 years old, no other seizures. He was on Ritalin, so it came to the Ritalin cause a seizure, or was it something else? And so, they want children, so I'm left with this dilemma, and she said, there's something there, but there's not enough data there to justify having him, tested. So
Yeah. So I think you bring up a lot of points that I can expand upon. One would be to say that that your journey is kind of what's happening now. So when I first started taking care of patients with epilepsy 14 years ago, we didn't have the ability to test anybody for anything. And then we had one gene, and we could test everybody for that one gene, but we didn't know about any of these other things. So in the last, 15 years, 14 years, we have now learned about 4,000 genes that we can test for that are related to epilepsy. So now we have a whole set of patients with epilepsy who are living out there who never had access to genetic testing as they were growing up and going through their diagnosis. And we didn't even understand how much a role genetics played in epilepsy at that time. And now we have a whole different perspective. So we're training people now. I mean, I'm so pleased by the training that I'm part of, because all of my residents know all about epilepsy genetics, and they know when to test kids, and they know when to offer it, and that is going to change the journey for all the people that came behind you. But what we need to do is catch up with all the people that are out there that never got that access, and that's why we just keep saying it's important for adults to get tested, especially if they had onset of their seizures at 3 years of age. And so I think that the adult epilepsy community is slower to catch on than we are, but they need to kind of participate in this effort, because I think what helps us is now she has this diagnosis that's very rare, and she's in her 30s, and all those other families who are just getting that diagnosis, they want to know what's going to happen to their kid when they're 30, and so she can provide them with that information, provide us as researchers with that information to help us to understand and do better at taking care of patients. I think another point that you brought up is the implications for other members of the family, and so maybe let Katie kind of field that question about what we would what we think about that.
Hey, let me ask you this then too, because when she had the genetic testing, I came back as having the same gene mutation, but I do not have seizures, and he came back fine. She also came down with something called ETE, which I think is more familiar. They test all the neonates now, when they're born for that, and if they're put on that supplement biotin, then it takes care of it. But she wasn't tested then, and I don't have that biotin deficiency, but that's something that came back as a pathogen.
Yeah, and I think maybe other people here have been in that situation. Variants of uncertain significance are really tricky, and it's something that we deal with all the time, is how do we tease apart what is meaningful and what is maybe just a normal change that runs in the family. And I think until we get to the point that we understand everything, or at least more, about epilepsy genetics, it's really hard to find those answers. We've come a long way, but we know that we have 22,000 genes. We know that there's about 4,000 that have some relation to epilepsy or neurodevelopmental disease, but there's still a lot that we have left to learn. So when we get a result like that, where there's an uncertain variant, we don't know, is there something else that's actually the answer, and this is just something that's a little funny and we don't know exactly what it means. And when we're talking about testing other family members, especially if it's going to impact how they manage a pregnancy, if they choose to have biological children, if it's going to change things like that, we want to make sure that we're testing for something that's really truly the answer, and not just something that happens to run in the family. So something like that you have the same change, you don't have the same symptoms and the same history. The question is, if your son has that too, what does that mean? Because if you have it, 50-50, your son has it too. Then that doesn't necessarily mean that it causes seizures or has the ability to cause seizures. There's just so much that we have left to learn. So I think that's why when we see things like variants of uncertain significance, we hesitate to recommend making big decisions, especially for other family members, based on those uncertain, kind of fuzzy results. I don't know if that really answers your question, but... Go ahead. Oh.
So I have a quick question. My son has trisomy 3p, so not an epilepsy gene, so to speak, but thought to be pathogenic. Can you just talk a little bit more about that? I've never quite understood.
So I think we can chat afterwards if that's okay. I'd have to look at kind of specifically, especially with duplications. It can be tricky because we need to know what genes are involved. There's a translocation. Okay. So yeah, with copy number variants of any kind, sometimes with duplications or deletions, we're talking about a whole chromosome, so a very, very big package of genes, and sometimes we're talking about a very small region. So when it comes to copy number variants, it matters a lot what the specifics are, and I'm happy to chat with you. I think Dr. Park had some thoughts.
I would also say it sounds like it's trisomy 3 or 13P, right?
Three.
So is it
3.
Is it 3 or 13? 3. And so it's the whole P partial. So I think that goes to Katie's point, but the in general, if we think about that category of things, the issue is that large packet probably contains genes that affect how the brain works. And so even though it isn't necessarily always associated with epilepsy, it can probably be because maybe the genes in that region are brain genes. And many of our genes have this kind of Goldilocks phenomenon to them that you have to have exactly the right amount of expression. You can't have too much and you can't have too little. So they require precise balance. So having extra can be a problem. And so I think that is potentially why there is an epilepsy associated with that, even though it doesn't maybe present with all of them.
The trifecta of autism, epilepsy, ADHD, like you always see the trio. But this
Yes.
doctor definitely thinks that it is causing that. But do we repeat it every year to see if we learn more, or not yearly, but I mean, what is the cadence for that?
Yeah, so when it comes to like genetic testing, you wouldn't need to necessarily repeat it. If they think that this is the answer, that's not going to change over time, right? So what we understand about it is what's always improving. So I would say anyone who has an uncertain genetic diagnosis, it's important to kind of touch base with the physicians that you're working with, the genetic counselors, whatever team kind of ordered that original testing. We want families to be checking in about what's understood over time. Some labs are really good about kind of initiating that process themselves. They'll send out updated reports when there's new findings. Sometimes it has to be family initiated or physician initiated, looking for those updates. So it's always important to kind of have that conversation. I would say generally on a yearly basis at least, so that we're prompted to see if there's been new publications. Have we learned more about the specific genetic change or variant that we're talking about? Those things are usually always getting better over time. Go ahead.
I'm wondering if you guys have noticed whether insurance companies and resources available to patients with these rare gene disorders have started to catch up in what they will cover and provide for these patients. Because for instance, I had a hard time getting my daughter into ADA therapy because she didn't have an autism diagnosis, but nobody evaluated her for autism, and then she got the autism diagnosis, and now we're getting it. So, just sort of like what insurance companies are willing to cover, because these diseases are so rare, and they've never heard of them, and they don't know what's going to work.
Yeah. So the question is whether insurance companies have kind of caught up over time to providing access to testing and therapies. I think we wish that they would more than they have. It can be a challenge. A lot of the insurance companies have kind of developed policies that are specific to certain types of testing, but they've kind of skipped over a lot of the testing that we do on a regular basis. So there's a lot of insurance companies that have policies that apply to single gene testing and that apply to whole exome or whole genome testing, where we're looking at everything. But they've kind of skipped over those policies for the panels in between. So things like epilepsy panels that we send a lot. We have a lot of trouble getting those covered. So it's a big challenge that we run into a lot. We fight with insurance companies a lot. I don't know if there's a good answer. We wish it was better.
I would agree with all of that.
Yeah.
I would also say sometimes having a precise diagnosis, like a genetic diagnosis, does give you access to things that you might not otherwise have if somebody just has a diagnosis of epilepsy. Because I think then people think, oh well, you might grow out of that, and or that might change over time. And knowing that in fact it has a genetic underpinning, and that underpinning can be associated with those other kind of attached conditions, can be helpful in some ways in dealing with insurance. I would also say that that another thing is about therapy is that therapy can be very challenging for our patient population. I think insurance has this view of therapy as, well, you had a knee replacement, and so you will need to have therapy for 2 months to recover from your knee replacement, and then you'll be done with therapy. And that's what therapy is, right? You had a car accident, you broke your arm, you will have therapy for however long that takes to recover, and then it will be done. And that's really not how therapy works for children with neurodevelopmental disorders. Children with neurodevelopmental disorders need therapies all the time, chronically and long term, to help them to gain skills and then to maintain those skills, and not just skills, but the kind of tone to prevent things like contractures and falls and balance issues from developing. And that perspective is very different. And so I think a lot of times what you need is an advocate to make that point and make that argument to the insurance and stand up for you in pointing out that this is a genetic diagnosis that is not going to change. It is associated with these things, and these things require chronic and lifelong management in this way. And I think that's really the case that needs to be presented. Yeah.
You mentioned that you can pay for the genetic testing yourself, but insurance doesn't cover it. About how much is that?
Yeah, so I think it depends on the test. So it depends on what's being recommended for you or someone in your family, and through what company you're using. So some companies have much better, kind of patient self-pay costs than others. I think maybe Katie can speak to a little bit more, or we'll be willing to talk about it afterwards. But I think it's very dependent upon what is recommended to have happen.
What about these genetic testings that are quoted on the hospital that you can see that you can pay, or less expensive, I think $400,
So like
that's
23andMe,
yeah.
things like that. So all of those kind of online tests that are out there, there's a lot of them these days. We call that direct-to-consumer testing. It is not something that we generally recommend doing. And the biggest reason for that is that type of testing is typically targeting common mutations and not looking at everything. So it's kind of like they're taking this whole encyclopedia and they're just like flipping through a couple of pages and picking out some main points, but it's not looking at everything. So especially in kind of the setting of epilepsy genetics, where a lot of those diagnoses are in genes that we haven't known about for a long time, they're very rare, they're very specific changes, that type of testing is going to miss things like that. So I think we advise against it in general if people are doing it for the purpose of learning about their health, because there's a lot that's being missed. So we worry about kind of a false sense of security if you're doing testing to screen for, family history of cancer, a family history of heart disease, family history of epilepsy, and you're getting that testing back and it's negative. It's not a very meaningful negative because they're not looking at everything. They're reasonably good at the ancestry side of things, so if you're doing it kind of for fun and for those types of reasons, it's kind of effective. But for health-related purposes, it's not something that we generally encourage people to do. Any other questions? Yeah.
Do you have any good patient success stories of using that precise treatment based on genetic testing?
Anyone that come to mind in particular?
Do you want me to, I can tell them the Mila story? Do you want me to tell the Mila story? Or should I tell a different story?
I don't know.
There's a publicized story about a young girl who had quickly onset of epilepsy, in addition to loss of developmental milestones, loss of vision, clinically got a diagnosis of Batten's disease, which is a neurodegenerative condition. They did genetic testing, and that disease is recessive, meaning that you have to have 2 copies of a gene not working in order to present with symptoms. And they were able to find one of those genetic variations, but they were not able to find the second one. And so that family partnered with Boston through, kind of windy road that included, Facebook and so forth, and ended up being able to do some research testing and identifying this second gene. And that second gene could be treated with one of these ASOs that we talked about that was actually specifically engineered for this young girl, and went through this huge approval process, being shepherded through by Dr. Yu at Boston, and was able to be made just for her. And it actually did help for some time to slow the progression of her disease. Unfortunately, it did not stop the progression of her disease, but it slowed it. And that family is now taking up her cause to open up that type of genetic testing for other patients, and doing research for patents and for ASOs in general, and I think kind of leading the charge. So it's a very well publicized story, and we were privileged at Children's to be part of it, and I think those things are increasingly happening. All right, I think we're almost at 11, so thanks for attending and for your great questions. And we'll stay and hang out if people would like to ask some additional questions in private, or touch base with Katie, who's kindly offered to speak with some people about their particular questions. And then we'll be out at the children's table if anyone would like to stop by. We're there all day today, and we'll be at the park tomorrow. So thanks so much. Bye.
Okay.